Hi everyone, thank you for coming. I'm going to talk to you about our new enzymes and our new product as well. So I'm going to introduce you our Solis Hot Start HiFi DNA Polymerase Kit, as well as our new master mix for it. In order to understand more about the high-fidelity polymerases, I will make a brief introduction into it as well. There are many families of prokaryotic and eukaryotic DNA polymerases, which are classified based on the sequence homology and crystal feature. Most commercially available DNA polymerases fall into one of the polymerase families, family A or family B. So the family A polymerases are usually DUC polymerases, whereas the family B polymerases are usually PFU polymerases, which are originated from Pyrococcus furiosus. So when we talk about the key features of these polymerases, then for the DUC polymerase, it includes the 5' 3' polymerase activity, as well as 5' 3' exonuclease activity. At the same time, the PFU polymerases have also the 5' 3' polymerase activity, and also widely known as the proofreading activity, the 3' 5' exonuclease activity. Mostly commercially available are the family B enzymes, such as the PFU polymerases, especially due to the proofreading activity that they have. This allows them to have more accurate amplification, which allows to make longer fragments due to higher precisity. Popular examples besides our Solis HiFi are Q5 from NIP, Kappa HiFi, Fusion, etc. And they can be used for any regular PCR work. There are also Phi-29 enzymes, which are mostly used for isothermal reactions, such as MDA and RCA. The general application areas are usually characterized by the one critical feature, which is more accurate amplification of DNA. So the high fidelity amplification part here. One of the largest application areas is definitely sequencing of the DNA or RNA. Where the accuracy of the DNA amplification relates directly into the quality of data. In addition to sequencing, there are many other applications where high accuracy of PCR is critical. For example, those applications where it is really, really important not to repeat the experiment too much, and where having exact sequence is really, really important. So, for example, any protein expression and production, also site-directed mutagenesis, also CRISPR-Cas9. However, the HiFi polymerases can be used when regular PCR methods or products don't work that much or as well. And these can be the cases for difficult templates or long templates. So HiFi polymerases can work with difficult templates usually better. So the sequencing market is definitely rising, and there will be increasing demand for this type of highly accurate PCR, therefore the HiFi DNA polymerases as well. Therefore, I am introducing once again our Solis Hot Start HiFi DNA polymerase kit. The enzyme itself is an in silico designed polymerase, and it has enhanced processivity and proofreading capabilities. This kit is available in a 3 tube format, and it includes the enzyme, the buffer, of course, and the 10x version of GC enhancer. We also just came out with our master mix format for this enzyme. We carefully optimized it and tested it, and it comes in a 5x format. So it's really stable thanks to it, as it is concentrated, and it's overall a really good choice. Core enzyme properties for the Solis Hot Start HiFi DNA polymerase would be the 3' 5' proofreading activity, which allows the enzyme to have up over 100 times higher fidelity compared to the Taq polymerase. However, this DNA polymerase does not have the 5', 3' exonuclease activity, and therefore it cannot be used with any types of hydrolysis probes. It also has increased processivity, which allows one of the applications to be long-range PCR. So basically up to 22, 24 kb of DNA targets can be used. And this enzyme has a chemical hot start, which increases specificity and allows for accurate amplification of complex and highly multiplexed assays. Furthermore, we have made modifications to the enzyme, so it can incorporate uracil nucleotides and also tolerate them. This enables unique and epigenetic based applications, for example, such as bisulfite sequencing. The enzyme has a stability tag incorporated onto it, which allows the product or the enzyme itself to be stable up to 1 month at room temperature. Thanks to this, this product can be shipped at ambient temperatures, as well as can be set up without ice. The DNA polymerase is really sensitive and stable. It has great sensitivity down to 1 picograms, while being comparable to competitors. As you can see here on the figure, here is the reference that was stored for 1 month at -20 degrees Celsius. You can see that the sensitivity is good. Here on the right-hand side, you can see the enzyme stored at this conditions. So for 1 month at 25 degrees Celsius. And you can see here that the stability has not changed. So once again, this stability tag enables the long stability at room temperature, which makes it really convenient for reaction setup and really good for environmentally friendly shipping as well. So the HiFi master mix has 24 hours of benchtop stability. What does it mean? It means that if you mix the reaction all together already, so with the targets, with the primers and everything, then it is okay to be left at room temperature or at the shell if you forget it, for example, then it's okay up to 24 hours. You can see it on the figure as well that the sensitivity and the stability of the results and the mix itself don't change. Our DNA polymerase, as you know, can amplify longer DNA fragments. So it has increased processivity, and due to this, it is definitely best for long-range PCR in our portfolio. So before we had Fireball, which could do targets up to 3 kb, and our hot Fireball blend master mix that could do up to 5 kilobases. So this one, our Solis HiFi, can do up to 24 kb with simpler targets, and the master mix can do up to 22 kb targets. The DNA polymerase is definitely great at multiplexing, so we have tested up to 18-plex in our experiments, as you can see here on the figure as well. However, it has also been demonstrated to be over 18-plex. So, for example, 40-plex has also been demonstrated by our beta tester. However, some optimization might be required for multiplexing to work most efficiently. So in order to do that, you can just increase the enzyme concentration, which allows the reaction to work more better. So also our polymerase is really versatile. So what does it mean? That it means that it can do efficient amplification of both AT and GC-rich targets. So here you can see the results of 30 to 79% of GC content. It is especially good when you add the GC-rich enhancer or any other additive that can help with those harder sequences. And it amplifies both low and high GC amplicons with similar efficiency. We have also tested the master mix to be really versatile and flexible. So here you can see with different types of cycling programs. So one was made for GC-rich targets, and one is especially good with AT-rich targets. We tested 27 to 79% of GC content here. And as you can see, both of the cycling programs actually can manage both ends of the other side, so the opposite side. So this shows the universal usage of actually the both GC and AT programs, which gives us more confirmation that our mix is really, really well optimized and it works with different cycling programs, which are usually made for certain types of targets. Additionally, our master mix has been tested to tolerate common PCR inhibitors, such as tannic acid from plants, a lot of humic acid from soil, some blood markers like hematine and heparin, urea and bile salts from urine, and a lot of sample pre-treatment chemicals. So you can definitely say that our master mix is inhibitor tolerant and can be used with many types of samples. Additional features of the HiFi DNA polymerase and master mix would be that it tolerates eunucleotides, which makes it well suited for certain applications, such as bisulfite sequencing and different methylation studies. It is definitely important in case of epigenetics and can be used in cancer research as well. It can also be used with damaged DNA, such as formalin-fixed paraffin-embedded DNA. And if somebody might need the carryover prevention of the samples, so when they're working with high throughput applications, then with UNG and adding of the UTPs or eunucleotides into the mix, in case of using the DNA polymerase kit, for example, the safety and insurance of clear and good samples are definitely ensured. As epigenetic studies is a growing factor, then this bisulfite sequencing, which can be used with both of these products of ours, both the DNA polymerase kit and the master mix, it is definitely good to think about it, as only a few other enzymes on the market can be suitable for it. So you can definitely choose one of our products for your different application purposes. So our DNA polymerase kit comes with increased fidelity. It is good for long-range PCR and has great multiplexing capabilities, so actually over 18 plex. It has great performance with both AT and GC rich targets and can efficiently incorporate the eunucleotides. So it can be great in both bisulfite sequencing or any other epigenetic studies, and also preventing crossover contamination. It is definitely room temperature stable for up to 1 month, therefore can be shipped and set up pretty conveniently. And there is also glycerol-free version available if you might be interested in it. And about our Solis Start HiFi Master Mix, it has also good fidelity. It can do up to 22 kb of DNA, depending on the target. It has also great multiplexing capabilities and has universal performance with GC and AT rich targets. It has good DUTP or eunucleotide tolerance, therefore it can be used in epigenetic studies. It has really, really good inhibitor tolerance, and therefore it is really robust. It has exceptional benchtop stability up to 24 hours, and it's also room temperature stable for up to 1 month. So thank you a lot for being here, for listening. If you have any questions, please, you may forward your thoughts and the questions to us in case of technical questions to support@solispyridine.com. And if you are interested in getting a free sample, then you can write to orders@solispyridine.com and get the sample for yourself. So thank you for being here and listening. Goodbye.